A very good afternoon to all the participants and our esteemed speaker, Dr. Animesh Acharya. Let me first introduce our speaker to all the participants. Dr. Animesh Acharya is CEO and founding director, Omics Marker Limited UK. The title of his talk is Big Data Driven Medicine, First Step Towards Diagnostics. A brief introduction about Dr. Animesh Acharya. Dr. Animesh Acharya did his BTech Electrical Engineering from Marist, Itanagar, India, MSc in Bioinformatics, IBAB, Bangalore, India, PhD in Omics Data Analysis, Wageningen University, sorry if I have spelled it wrong, uh, the Netherlands. Currently, he is a senior scientist at University of Birmingham, UK. He is also CEO and founding director of Omnics Marker Limited, UK. Dr. Animesh is a visiting professor, NIT Calicut, India, and a visiting scientist, University of Cambridge, UK. His research interest includes omics, data analysis, feature or variable selection, and data integration or fusion. Now, I would like to kindly invite Dr. Animesh Acharya to hand over the session to him and carry on with his talk. Thank you, sir.
Thank you very much. Firstly, I would like to thank the organizers for inviting me, and especially Professor Rajiv De and Dr. Rishamal. It is really a privilege. Being in UK, I can deliver some of the lectures online nowadays. First of all, also, I'd like to thank other people involved in this colloquium and in this program. So, as the title says, the big data-driven medicine, a first step towards the diagnostics. So you can see there are a couple of key words in this title, and the big data becomes quite a happening word nowadays in the industry and as well as in the academic settings. Now, because my appointment is a kind of a industry academic setting, so I'm not I have to focus on a little bit of both the side, but maybe I'll focus on some of the research I'm doing in the academic settings. But they are very translational aspects of it. So, so the agenda. So I like to cover a couple of things for today's meeting. Is number one, the introducing the big data. The second is identification of the biomarker. I mean the big data and biomarker and diagnostics, somehow they are linked. And I would like to establish the fact that how this big data is linked with the biomarker. I would like to show some of the case studies, and those are the case studies is mainly in the interdisciplinary settings. And then I would like to focus on how people can do research in a very multidisciplinary or interdisciplinary framework. So which is very important nowadays. So let me start with the first slide. So which is the topic is a big data. So if you talk about the big data, the first thing is coming to your mind is how you define the big data. And I do get these questions in many presentations. I mean when you say data is become big, so there is not any specific definition of the big data, but the big data is a maintaining some sort of property. So one of the property would be there will be very high volume. So maybe you cannot express everything in your desktop. So you need some sort of infrastructure. And they have quite a lot of velocity in the sense there may be the real-time data. So you can get an enormous amount of data in a single shot. And then there are other properties like veracity and variety, different types of data, and those are the 5 different aspects is included in the big data definition in a way. Now, the big data I'm talking about here is mainly about the biomedical settings. So if you look at the right hand side, you can see how people are This is the first paper came out in Nature when human genome sequences started. So approximately 20 years back from now. So around 2000, when first human genome sequences started, then as a consequence of human genome sequence, they become a source of many different big data. And many papers like Science, Nature, they published the sequencing, different databases, and people started looking at the data in a multiple different ways. Now, if you look at the explosion of the data sets, and as this meeting is about the biomedical settings, so all the examples are taken from the biomedical settings. You can see this is the European Bioinformatics Institute, where they are trying to show how sequencing is because of sequencing of multiple different species, how the data sources has become enormous growth. So this is quite old chart, but nonetheless, it gives you some sort of impression how the data is becoming too much and exponential growth of this. Now, if you look at the other things, like for example, Twitter and other social media, which is very active nowadays, you can see how many the types of data you are getting is also quite enormous. You talk about the GPS system, and you can see that they are all real-time data, and eventually it becomes a source of the big data analytics. And there are many other examples. So the other examples, you can see the guy with a drone. So this is the data is coming from the plant science, and this is from the Colombia. So this Colombia, I have some collaborations with Colombia. So we are working with them on the data is coming from the drone, automatic plant phenotyping. So they wanted to reduce the number of people working in the plant. So they made a huge trial of the cassava root and cassava plants. So they wanted to measure the plant height, the plant leaf area, and all these things. So they are using the drone, but then the entire thing need an infrastructure. So drone giving you pixels needs to be done as an image processing. After image processing, generation of the quantification of the data, and then analytics coming to the picture. The third picture, I'm showing the MRI or ECG MRI screen, where you are brain scanning. In the brain scanning, you do get also different scanning, and then you wanted to make sure how this data can be analyzed in the setting. So these are all kind of a source of examples, some of the examples of the big data analysis and big data sources. Now, looking at the literature, if you look at the literature, you can also see there are quite variety of the articles. For example, the first one is the Journal of Big Data. You can see in the healthcare domain how people are emphasizing the data sets and the problems, management, and the limitations as well. Yeah. So I think this came out in 13 January in the Nature Medicine, Big Hopes for the Big Data. So if you get a chance to look at those papers, you can see how people are looking at the data, but not only they are appreciating the big data, but also there are some of the holes which we need to answer as well. So big is not always mean it will be a resourceful. Big always sometimes can be meaning is a chaotic, more chaotic, and more possibility you are landed up with the false positive results. Yeah. So those are the challenges you can also see in the many important articles like Nature Medicine, Cell, and those type of articles. Now, as you can realize, this big data is a very broad term, and then given your situation, you can contribute in multiple different ways. And some of the aspect could be the image analysis. So, for example, in the biomedical engineering settings, or electrical or electronics engineering settings, even computer science engineering settings, you can see image analysis can be one of the domain. Natural language processing, artificial intelligence, machine learning, statistics and mathematics, and knowledge representation. So knowledge representation, I'm talking about mainly on the ontology and how do you represent and the problem. So based on the expertise, so people are coming from multiple different domains to make it happen, and then becomes a very interdisciplinary source of area. Now, the group I am in, so we call it the Clinical Bioinformatics Research Group, and I am heading a subgroup of this huge group. So we are part of Alan Turing Institutes in UK, where Alan Turing Institutes encourages a lot, like machine learning, and theoretical computer science. I'm also part of the CCB, Center for Computational Biology. I am also affiliated with University Hospital. So one of the advantage of affiliated with hospital is you get a lot of clinical trial data, yeah, and closely working with the clinical people. 2 years back, not 2 years, 1.5 years back, government of UK started new thematic area called HDR UK. So that is called Health Data Research UK. So UK government is spending quite a lot of money on the health data research. Now, below you can see there are thematic areas. For example, comparative genomics, large-scale representation, harmonization, actionable AI, network medicine, translational medicine, symbolic artificial intelligence, multi-omics integration. So these are the group. We are around 22 people in our group, and heading by Professor Georgios Goutos, who is the director, one of the director of the HDR UK, and I am leading omics analytics group. So my group consists of around 6 people, consists of 3 PhDs and 2 postdocs and other technicians. So I'm more in the area of biology and AI is a mix of together, not completely AI, not completely biology, but mix of both the knowledges. Yeah. So I'll be talking about more on the omics or biological insight and the fusion of the machine learning techniques in the current slides. So if you look at the any sort of the data, doesn't matter where you are, you will be working in any of the slides. Okay. So the lower slide is talking about the genome and from the human side, and you can see this is the different layers of the organization of the molecular settings. Yeah. So genome, transcriptome, microbiome, metabolome, and then ECG. So ECG is a phenotype. You measure ECG as a clinical phenotype, clinical outcome, MRI, exercise, retina, clinical letter, electronic health record data. So these are representation of multiple different data sets. So we can classify these two data, this kind of organizational of molecular organization in 2 different areas. One part is omics analytics, and I'm going to describe what does omics mean in coming slides. And the other one is the phenotype-based analytics. Now, the phenotype-based analytics, what does it mean? The phenotype-based analytics mean is like what you can measure. Yeah. So, for example, ECG, MRI, or maybe you are measuring the glucose content of the patients, or maybe the prescriptions, or we call it electronic health record. Those sort of settings. So for today, I'm focusing, as I said, mainly focusing on the omics type of data sets, and just to give you an impression how omics can merge with computer science and mathematics and statistics, and give you some sort of the outcome. Okay, now that was the little bit of background. Now, if I wanted to describe the system, I know the people who are in the electrical engineering settings, they know this very simple diagram very well. So you are giving an input and some system you are considering as a black box, and then you are getting output. Okay, and then it might so happen you are getting some feedback. All right, so that can be as a based on your input and output, it can be the a single input, single output system. It also can be a multiple input and multiple input, multiple output system, based on the system, of course. But this is a very a simple representation of any sort of system. Yeah. Now in biology, it happened exactly similar way. So we call it the input as a genotype, and we call output as a phenotype. Okay. So the genotype is which you are, some part of the heredity based on your sequence in your DNA is mediating across multiple different progenies. So that is a part of the genotype. We call it natural variation in your genes, natural variation. Yeah. And then you measure the phenotypic characteristics. So as I say, genotype refers to the genetic blueprint encoded in the DNA, and the phenotype represent the observations and the physical manifestation of that information. so you can think about it's a kind of a very naive way of looking at a control system, actually. So you probably already guess what I'm trying to say in terms of the engineering point of view, system engineering point of view. And then it is also happening. If you can control of the some parameter, you can also control the output. Okay, so you control the input, you are controlling the output. Now, problem becomes if you cannot control the input and the system become divergence, then your phenotype is not controlled, and it becomes would be which parameter you want to control, which one to target. So that is a very critical steps in the biology. Okay, so um, so in a way, you can see that, and then it come up the omics kind of system, where omics suffix is a multiple different feature or a set of features. So if you are looking at the genes, you can say genomics. If you are looking at the proteins, you can say proteomics. If you're looking at the metabolite, you can say metabolomics. And these are all quantitative. These are all quantitative, massive amount of data. Okay. Because their numbers of features are quite large. Now, you can see a small box here, and what I'm trying to say here, you give your input as a genotype, and you gave your output as a phenotype, and this is not exactly a very simple thing. And then what is happening here is a little bit more complex. So you have a DNA, and then it is going to RNA, mRNA, and proteins, and each of the steps we can measure nowadays. We can measure how much DNA is transferred to the RNA, how much RNA is transferred into mRNA, and then with the feedback. So it becomes a very complex control system in this case. And controlling is happening each of the steps. Even you don't know where is the controlling is happening. And the problem is control is happening in the micro level, very, very micro level. Yeah. And because of this, you get a different sort of disease. Some of the diseases are genetics. Some of the diseases are environmental, like diabetes. Yeah, so in diabetes is a multifactorial, like your age is responsible, your healthy system, how do you behave, you are exercising, and all these things are is also responsible. Yeah, so it depends. But what all I'm trying to say, number one is we can measure how much gene is transcribed. We can measure how much protein is transcribed, and we can identify if the proteins or if the transcripts are important for some of the diseases. Now, if you look at the omics analytics, in the omics analytics further, further big data can be arising, and then one of the aspect is the biomarker and diagnostics. Yeah. So this is a really a subdomain, if I can say. This is really a subdomain across many different domains. And we are doing quite a lot on the biomarkers and diagnostics. Even my company is doing some sort of diagnostics measurements. The second aspect is the workflow and a framework. You need a workflow. You need a framework to systematically identify those. And then third step is the causal modeling. So causal modeling means if A is causing B or B is causing C. Okay. Now I know the causal is a very hard, and in the computer science and theoretical mathematicians, I mean, their causality is a little bit different from the way the clinical people are defining causality. So the way we define causality is we do experiment. We do a lot of experiment to establish the fact that if some of the genes, some of the proteins are causing the disease or not. But of course, I mean, we use a lot of graphical models like probabilistic Bayesian networks and probabilistic graphical models to establish which can be the candidate. The fourth step is the stratifications, and stratification and diagnostics, they are going hand in hand. I mean, once you identify the diagnostics, and then you wanted to stratify your future population. Now, in this area, it is very important to describe the characteristics of omics, how omics data is looking like, and how different omics data compared to the normal data set. Yeah. Now, the omics data is a wide data. So what is wide means in this case? So you can see the left-hand side, I'm trying to project what is my Y. So Y is my outcome variable in this case. Now you can consider my outcome variable can be disease, non-disease, diabetic, non-diabetic. Okay, so this is a binary class. It can be multi-class. It can be the healthy population, cancer stage 1, cancer stage 2, and cancer stage 3, for example. Okay, so it can be the multi-class. It also can be, Y also can be my quantitative, okay, like age, BMI, okay, those can, or measurement, some of the measurement, like glucose concentration of a in the population. Yeah, so these can be quantitative. However, if you look at the predictor metrics, it becomes a mess. Mess in the sense, when you do an experiment, in a single experiment, meaning a single sample, you get around 50,000 features. Okay, some of the experiment landed up with 1 million features. Okay, you cannot even open in your desktop, so you need some sort of a cloud, or you have to do analysis on the fly. Yeah, so so that is wide. The reason it is wide, your feature space becoming so much, and your number of samples and number of experiments are not increasing that much. For example, how many patients you might get? 1,000, 2,000, 10,000, maybe 20,000, but your number of features are 1 million. So it's heavily underdetermined system. Okay, so that's the first thing. So if you wanted to make a solution of the system, you need to think about how do you make the some sort of a pseudo solution, or you need to think about how you make the identify your important features. Okay. Now, now I think I already explained here, and then you can see as a consequence, you become a very big data set. So in omics settings, the big data is not in terms of the number of samples. It big in terms of number of features you are considering. Yeah. Now, eventually, you are asking yourself, okay, which one to target? Which one I should go for? It is it I should go for 20? I should go for 10? How should I go about that? And once you identify which one to go about it, and then those guys, those 20 or 10 can go for diagnostics. So one of the first steps for diagnostics is identification. Yeah. So as I said, and we call it as a biomarker, okay, because they are marking something as a out of 1 million, they probably doing something with Y. Yeah. And that's why we can call it a biomarker. And if they're biomarker, we can take it one step further. We can say, okay, they maybe can be used as a diagnostics. Now there is a hole here. I mean, the question is, I mean, you cannot call everything as a biomarker. Okay. Now, if you want to call as a biomarker, there has to be 4 properties. Number 1, you should measure. The entity, the feature should be measurable. The feature should be indicative. Indicative means, in the sense, the feature will tell you the state of the disease. They should be predictive and robust and stable, and then you can call it as a biomarker. So, and it is very hard in this type of domain to identify which you call as a biomarker. So this is really a important aspect, but in the same time, it is very hard to achieve that. Now, if you look at the literature, I mean, people used these kind of terminologies in multiple different names. So they call it translational biomarker, drug response, genomic medicine, precision medicine, personalized medicine, population-based stratification, predictive medicine, many different names. But actually, if you look at carefully, they are looking for those sort of properties. And once those sort of properties are established, then you can go for the diagnostics. Yeah. So what I'm trying to say, you have a big data, identify the features, claim their marker, and then go for the diagnostics for stratification. So clearly, you need a kind of a framework or some sort of a workflow how you are making those links. So here you go. So if you wanted to make a link between the biomarker, it has to be a process-driven approach. So, for example, given in your data, you apply some sort of method, and I use quite a lot of method, starting from artificial neural network, support vector machine, deep learning, depends on what method. So I run many and let data decide which method can be useful, and then you fix the method, you identify your important marker, and you generate new data. So that is very important step. So generate new data, in the sense we call it test data, because this test data is will be used for the future, predictability. So these data I have not used training my model, and I am in a position to see how those markers are performing as a new data. Okay, so we do a trial, generate a new data, predict, and quantify. We do quantify as a area under curve or ROC curve, or if it is a quantitative Y, then we say how much variation is explained. So this is the work for doing by my Laura. So Laura is my PhD student from the biomedical engineering, actually, and she is working heavily in this area, developing the workflows and framework for biomarker discovery. So in a nutshell, so we started doing it. We started doing some part of it. So and for example, in the left hand side is a you can see the raw data is coming, and each data has got any different sort of like missing values, outlier detection, and normalization. So we made one module there which is taking care of that, and then process data is going multiple different methodology. So we use in this case we used actually a Lasso and elastic net. So Lasso and elastic net is motivated by the linear regression. These are high dimensional statistics, and you can see we also call it as a penalized regression. So idea of penalization is the features which are not doing anything, just sitting in the data, they automatically kicked out from the model, and that's the way we do the solution of the system. Okay, so you realize this is a very heavily underdetermined system, P greater than N problem, and using those methods actually helping you to get a estimate of your features. Now, I have to say that those estimates are not a real estimate. They are kind of a pseudo estimates. Yeah. So because you don't get a real solution of this kind of system, but you need to solve the system of equations. Yeah. So we do a tenfold cross validation to optimize lambda. Lambda is a penalty parameter. So you give a more penalty to the more penalty, you get more penalty means heavy penalized. You get less features in the solution. You relax the lambda. You say, okay, less penalty. You get many features in your solution. Okay. So we published last year, and this paper, and what we showed in this paper is we showed multiple different big data. We took the data from the public resources. We showed this framework is robust. This framework take into account the stability. This framework take into account reproducibility and quantification. And we compare the results in the literature, and we claim that this kind of framework is necessary and can add value in the literature by adding reproducibility. So this is we published in Journal of Translational Medicine, and then exactly the same framework we applied in the trauma. Okay, so what is the trauma? So as I said, my affiliations with University Hospital, we get and UK National Defence Center is in Birmingham. From UK, people are going for fighting in Afghanistan and Iraq, and they come back in Birmingham Hospital, and they are trauma. They are blasted in the fight, but also, for example, car accidents or people fall over from the height, accidents, major trauma. They come to the Birmingham Hospital, and what we do, we took the samples, blood samples, and the serum samples and plasma samples, and then we do a lot of experiments. And one of the questions we are asking is, can we identify multi-organ dysfunction? Now, what is multi-organ dysfunction? So multi-organ dysfunction means when people get a trauma and your entire system, your body homeostasis is disturbed. After 5 days and 2nd days, you may find your kidney is not working. Okay, and then you found your liver is not working. So these are the sequential process of damaging your organs. For example, your pancreas is not working. So they are sequential organ process, and because of the sequential process, you get a sepsis. And then once you get a sepsis, there is no way you can reverse back the patient. That's why there is no cure for the septic patient. Septic patient will die because of your body is your cytokine storm in your body is so much, and it doesn't work. So we have to identify before. We have to identify before someone is getting MOD or septic. Okay. Now, question we are asking: can we identify MOD? Which patient is going to MOD? Which patient is not? If we can identify earlier, then we can take care of those patients in better way. Also, we can take care of this patient, which specialty hospital we wanted to send. Is it Birmingham? Is it London? Or is it somewhere in Oxford or maybe Manchester? Where do you want to send this patient? Yeah. So we applied this sort of thing, previously published data on cytokine in the PLOS medicine, and we took those data. We took this data, and this data is a longitudinal data. So you can see here trauma, and then here less than 1 hour sampling. So people got a trauma within 1 hour. We do the measurement, and then we did another measurement 4 to 12 hours. We did again 48 hours. So we wanted to see if there are any cytokine, if there are any inflammations are predictive in less than 1 hour, and 4 to 12 hours, 48 to 72 hours, but also how the dynamics of those cytokine profiling, how they are mediating across multiple different time frames. Okay, so you can see here in the different, and we identified selected features. So selected features in the left hand side, you can see the AUC values, so quantifications of how much predictive each of the marker. We identified the monocyte is important, and CD63 and CD62. By the way, these are all the clinical omics features, so you don't have to remember that. All I'm saying, we found some signatures in less than 1. We also found some signatures in the middle time period and then later time period. And if you look at the box plot, you can see how the monocyte is fluctuating. So for example, you can see the patients which have a no MOD and MODs, they kind of initially their variation is quite high in the first, and then their dynamics are kind of settling down. Yeah. So these are very interesting. So monocyte could be one of the target, and we are doing now experiment to prove it, how it is behaving. Yeah. So the message is each of the time frame, maybe they have a very specific marker, but dynamics of the markers is important because your body is changing in the trauma, and your marker concentration is also changing accordingly. And we published this data last year in Nature Scientific Data, and if you read it, you can see we actually applied a sophisticated framework in this sort of data. Okay, and then we are now working on the complex framework, how we can include neural net, support vector machine, and all these type of things for the translational medicines. Okay, so this is one case study. The second case study, I'm talking about the microbiome-based diagnostics. Now, this is the work for my master student, and she did an amazing job. So basically, she's from India, she's from IIT Delhi, and done mathematics and computing dual program, and now she's learning biology. So that is amazing combinations. I mean, people knows equations, people knows the computation, people also know now she's learning the biology. So it's really amazing, and we need this type of people in future. So microbiome-based diagnostics. So microbiome, these are the bacterial species staying in our gut. And research, last 10 years of research has been found, those are amazingly notorious. And they are involved any disease, starting from your Alzheimer, dementia, starting from cancer, diabetes, NALFD, non-alcoholic fatty liver disease. They have heavily involved. Okay. And here in Birmingham, we are also involved called FMT study, FMT called fecal microbiome transplant. So idea is you look at the fecal samples, healthy fecal samples, culture which is good bacteria, and cure the ulcerative colitis or inflammatory bowel disease. Sounds very ridiculous, but it's happening actually. We have the protocol to do the experiment here. Okay, now if you look at these diagnostics, what is happening? These are the very high impact journals. So for example, the commentary recently got the microbiome-based companion diagnosis no longer science or fiction. So these guys claiming it was a fiction some point, but it's no more. Okay, this came out is a BMG journal, which is impact is nowadays 22 impact factor journal, the highest in gastroenterology. Microbiome-based diagnostics ready for application. So these are the articles people are criticizing. Also, people are appreciating the fact that maybe microbiomes are one of the way to go for many different diseases. And in the lastly, you can see the liver fibrosis. I mean, if you are drinking a lot of alcohol, at some point your liver will give up. Of course, if you also don't drink alcohol, there is non-alcoholic fatty liver anyway. So liver fibrosis, and then because of this, can some microbiota is involved, and this came out, I think, from the United States in Salk Institute, but quite interestingly. Okay, now, so what I'm going to say is we have been working on the microbiome, and as I already gave you a little bit of introduction of the microbiome, and you can see they are everywhere. Metabolic diseases, they are in the cancer, gastroenterology, but also a lot of neurological diseases like dementia, Alzheimer diseases. Even they are linking the gut microbiota axis. So from the gut, they can go to the brain and manipulate human stress and other things. Okay. Now, these are the very recent literature review, and you can see again, this is just a summary of what I said, justification of what I'm saying. Many papers, you can see those papers are very high impact papers. Cell metabolism is 22, Nature Medicine 30 impact factor journals, and they repeatedly saying the same aspect. Now, the important part of the microbiome is microbiome is little bit different types of data. So I am trying to show here a very brief snapshot of the microbiome data, and immediately you can see the problem. The problem is you can see the values. Each of the values in the Excel sheet, they are very low, 0.0000 some values, yeah, and you can also see very zero values, so sparse, yeah, and then in the column, these are the name of the bacteria and enterobacteria and all these bacteria, yeah. So we call it taxa and ecological covariate kind of thing. But mainly the problem is sparsity. Because of the sparsity, we cannot apply the simple linear modeling here. Collinearity definitely a problem in this type of wide data, nonetheless of microbiome data. But I hope you already got if your matrix is p greater than n, by default you will get a collinearity. So you cannot invert your variance covariance matrix. Your beta is not estimated. So the collinearity is a problem. Effect size. So microbiomes are not working alone. So they are working as a colony. Okay. So if they're working as a colony, then what is happening? Each of the effect of the microbiome is additive. So they added up the effect and doing something in a colony. Yeah. So these are the three problems actually we wanted to address here. And if you look at the literature, and these are the very good papers. And then we did some sort of a review where people are doing what, where people are focusing on upstream analysis. So what is upstream means? You do experiment and then you do quality control of the data. So that's the upstream. And then once you get a quantification, you do a downstream analysis. Yeah. So many people say this is a problem in the literature. Yeah. So we wanted to make a framework. Our objective is we want to make a framework and trying to develop a novel methodology. So and she did an amazing job. She developed a methodology. We call it NFNet-FU, which is a neurophagy network fusion. Okay, it sounds very very sophisticated, but it is actually. And I'm going to tell you a little bit on this. So these framework can take into account sparsity, effect size, collinearity, heterogeneity, and then the final step, we introduced a lot of biological data in the top of it. Yeah, and we applied 3 different data sets. So these 3 data sets have been published, either mouse data. You can see the 2020 data, 2015, 2016. So these are the already published data. We applied and we showed that this framework is making sense and reproducible. Okay, so this is the schematic diagram of the pipeline. You give an input, and we modularize different aspects. So for sparsity, we use adaptive neurophagy inference system. For collinearity, we use an unsupervised DB scan. Some people so the unsupervised some PCH sort of analysis. We use adaptive Lasso. for feature score. And then in the fourth module, we combined biological information or the clinical information, microbial diversity information in the enrichment analysis, and we developed a new score. So based on the score, you can prioritize which microbiota is you are going for the translation or diagnostics. Okay, so that is the added value of this framework. And then just to show you what is happening, you can see the left-hand side figure is very, very sparse. And once you apply your rule-based method, neurophysics system, it completely transfers the data in a high-dimensional space and some sort of numerical value, and you don't see any sparsity. And you do the calculation in the high-dimensional space in the rule-based settings, and then you transfer back. Yeah. So that's the whole idea. So I'm not going to discuss in detail because I'm probably run out a little bit of time. But what I'm trying to say, we address individually. We address collinearity in using the unsupervised machine learning technique called DB-scan, and trying to identify which of the microbe is actually, or colonies of the microbiome is affecting our different diseases. And this is a kind of equations where we use the adaptive lasso. So one of the advantage, we have not used the original lasso. Original lasso is by Tipshirani in 1996. There are many variants of lasso, adaptive lasso, fused lasso. So one of the advantage of adaptive lasso, you can estimate the weights. Okay, so penalization not uniform across all your features. So you can weight which parameters are more weighted, which parameters are less weighted. So you can keep track on this. Yeah. So yeah, and then finally we develop, you can see the two networks or graphs. We use a lot of graph theory in this case. The left-hand side figure is a completely data-driven, and then we enrich the network based on a taxon set enrichment analysis, and we say, okay, look at the node 2. Node 2 might be interesting, and then can follow up for further validation or experimental protocol. Yeah. And we as I said, I mean, we before the validation, we also did some sort of literature-based validation. For example, here you can see there is a Prevotella bacteria is abundance we found in the cancer, and we're trying to show here that in the literature people talked about it. We are now trying to doing some NLP-based automatically what we get from the networks, do the NLP, get all the bacterial species in the literature which is associated, and fuse in the network and make a more weighted graph in this case. So yeah, so and then so in a nutshell, what we are saying is that we developed a novel actually methodology, and we compared with the literature data, and it seems working well so far. But of course, there is a room for improvement in all the research. One of the problem is the time complexity. Although we are using the cloud, sometimes optimization takes quite a lot of time. Even though I use the non-convex optimization, still it takes some time. But also we need to compare with other very comparable. Each of the module we have not compared. We compared overall, but each of the module has to be compared with the competitive what other people are doing here. So we submitted literally last week the genome biology, which is quite good paper in this, and we hope to get some reviewer comment, and then we'll modify it accordingly. So this is my last case study, and then I'll be very short, and then maybe we can discuss about if there are any questions or comments. So this is another case study. We did fusion. Okay, so far, if you notice, I was talking about only one set of data. So either inflammatory data, transcriptomics data, metabolomics data. So these are individual data, or microbiome data. The question is, what if you get a multiple measurement, a multimodality? We call it multimodality. So, and these multiple data sets are measured from the same samples, meaning same patients. Yeah. How do you handle this type of data? How do you do to identify your feature? How your diagnostics look like in this setting? How your predictability looks like in these settings? Yeah. Can you identify? Can you prioritize which data is important? So for example, in one of the study, we generated 10 different multimodal data, and our stakeholders wanted to know, okay, which one is more important? can you prioritize? So the idea is, if one is important, we can follow up study only for those. We don't need 10. Yeah. Okay, so that's the settings, and then in the settings, and you can see here there are quite a lot of literature actually in these settings, and then in the data fusion, people call it data fusion, people call it omics integration, people call it multimodality, you name it one, but they are essentially pointing towards the same thing. So these articles, you can see these articles sometimes looks like very, very biological, but believe me, they looks biological, but in the behind the scene, there are quite a lot of computation, machine learning, statistics, mathematics behind that. And then for example, BMC bioinformatics. This is the mathematics of the integration. In Nature Immunology paper, they talked about the cytokine, but they use the deep learning. Yeah, and that's why the impact goes up. I mean, if you do deep learning in a computer science setting, well, you can publish IEEE, which is 4.5, which is good, but if you do the clinical data, your impact, we are talking around 15 impact paper journal for sure, if you are using very sophisticated. Again, the nature communication type of papers, which is also quite good in this sense. Okay, now, so again, coming back to the IBD or gastroenterology, kind of a case study. So one of the diseases called the PSC. I mean, I'm sure nobody knows about it. I mean, it'll be good if you know already, because when I started this project, I didn't know about what is PSC called. Because being in engineering and settings and other things, how do I know that what is PSC means? The whole world was dark at that time. Yeah. So PSC is a progressive inflammation of fibrosis and biliary tree. Now, the same slide, this PSC is not exactly as IBD. So people, the clinicians, if you get a PSC, the clinician or GP will say, you are suffering from IBD. But this is the claim of the paper. PSC is not IBD. PSC pathology is very different from ulcerative colitis or Crohn's disease. Okay, so that's what the pathogenesis of PSC is poorly understood. And we wanted to identify which are the major players in PSC, so that we can do something around it. We can identify the drug target, and then accordingly we can work around it. So if you look at these data, so what we did, you can see the number of samples here, and I know some of the people will be already, and whenever I'm presenting this slide, the first question I get, number of samples, you are talking about 10 samples, and which is nothing, which maybe I'm looking at false positives. I agree with that, but then nonetheless, we are doing some validation experiment, and we are doing also a trial on this. Yeah, but this is a pilot study. So the mucosal samples, we did RNA sequencing, we did immunology, we did a microbiome, and we generated some of the matrices. So you can see these are from same samples. Again, these are the wide data in each of the in each of the settings. Yeah. And then we use the random forest decision-based algorithm here. We optimize the random forest method to identify the selection of feature, and then we synthetically made the graphs. For me, it is a graph. If you talk about with a clinician, they call as a pathway. Okay, so for me, it doesn't matter. I mean, as long as they are happy and I can communicate with them, that's all right. Yeah. Okay. Now, mathematical representation. So how do mathematicians will be representing this sort of data? So this is a mathematical representative view. How the mathematician? So mathematics, I mean, you will see. Okay, you have an X matrix, you have a Y. Y is same. You can see Y, same Y, because they are same patients. And then you can increase dot 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 X, Y, Z matrix. You keep on increasing the matrix. Doesn't matter for me because I made a framework. Each of the matrices will automatically reduce the dimension. So you can see the white, the dimension become dimensionless, meaning you have identified the features which are important, maybe 2 or 3, so dimensionless. You combine those features, do some sort of a graph, and you can see they are different color. Different color representation is they are coming from different data set, and then you